Welcome back and welcome to a new video on bacterial staining.
There are two major types of bacteria, and they are archaea bacteria and Eubacteria. Eubacteria are divided into two groups known as gram-positive and gram-negative bacteria. All bacteria can be stained using gram stain.
There are different methods of staining bacteria in order to study their structure, and in this class let us study some of the most common and most important methods of bacterial staining. The first and the foremost method is called gram staining of bacteria. It's called gram staining, and this was developed by Christian Gram in 1884. And this method is popularly used to differentiate bacteria into gram-positive bacteria and gram-negative bacteria. We will study gram staining in detail in the practical class, and I'll make a separate video on gram staining later, and I'll post it later. In this class, let us only know the definitions and the basics of different types of staining of bacteria. The first one, as I mentioned, is called gram staining bacteria. It's a bacteriological laboratory technique used to differentiate bacteria, as I mentioned, into gram-positive and gram-negative. You are seeing on the screen two types of bacteria. Gram-positive bacteria after staining will appear violet in color. Gram-negative bacteria will appear red in color. You can see here the gram-positive bacteria on the left-hand side, which are appearing in violet color because we are using crystal violet color. So when we use crystal violet color, the gram-positive bacteria will take up the crystal violet, they appear violet in color, whereas the gram-negative bacteria don't take the crystal violet color because of their cell wall structure. I'll come to that later, and hence we use a second stain, and the second stain that we are using is saffroning, which is red in color. It's a red colored dye, so that is why gram-negative bacteria will appear red in color. So gram-positive bacteria appear violet in color, gram-negative bacteria will appear red in color because of differential staining. The difference between gram-positive bacteria and gram-negative bacteria mainly lies in their cell wall. As you can see on the screen, the gram-positive bacteria have a simpler cell wall, whereas gram-negative bacteria will have a complicated cell wall. In case of gram-negative bacteria, we have two layers of cytoplasmic membrane. There is an inner membrane, there is an outer membrane. In between inner and outer membrane, there is a little amount of peptidoglycan. Whereas in case of gram-positive bacteria, there is only internal cytoplasmic membrane. There is no outer membrane. Cytoplasmic membrane is not there on the outside, but at the same time, there are several layers of peptidoglycan. So that is the difference between gram-positive bacteria and gram-negative bacteria. Because of this difference in their cell wall, gram-negative bacteria will not take the crystal violet stain, and hence they are appearing red in color because they take the second stain, that is the saffroning stain. Whereas on the other hand, the gram-positive bacteria will take up the crystal violet stain in the first state itself, and hence they appear violet in color. So that is the basic difference between gram-positive and gram-negative bacteria. Coming to the second type of staining is called acid-fast staining. The primary stain used in acid-fast staining is carbol-fuchsin, a lipid soluble and contains phenyl, which helps the stain penetrate the cell wall. It is a laboratory test that is determining the sample of tissue or blood and other body substances infected with bacteria that cause diseases like tuberculosis. So acid-fast stain is a stain which is used to stain the bacteria like tuberculosis in case of infected tissue, infected blood, or any other types of body fluids. So the purpose of acid-fast staining is to stain the bacterial cell walls to differentiate between acid-fast bacteria and non-acid-fast bacteria. For example, the bacterium that causes tuberculosis, that is Mycobacterium, has a cell wall made up of 60% mycolic acid, and hence it takes up the stain. and which is called adherence factor. You can see on the screen the acid phos negative, acid phos negative. You are able to see the differential staining of acid phos negative and acid phos positive. So this is what is called acid phos staining. And the third type of staining is called endospore staining or capsule staining in bacteria. So the cells, bacterial cells, sometimes form endospores. By using certain dyes or stain, you can stain the endospores. On the other hand, we have the capsule stain, where each and every bacterial cell has a capsule, and the capsule also can be stained. In that case, the bacterial periphery, the outline of the bacterium is stained. Some bacteria are capable of changing into dormant structures during unfavorable conditions that are metabolically inactive and do not grow and reproduce at the stage. Since these structures are formed inside the cells, they are called endospores. These structures have remarkable resistance to heat, radiation, chemicals and other agents that are typically lethal to the organisms. The heat resistance of these spores has been linked to their high content of the calcium and dipicolinic acid. So because of high content of calcium and dipicolinic acid, the bacterial cells in endospore conditions are highly resistant to external factors, and that's why they are able to survive even during unfavorable conditions. And we can use endospore staining whenever the bacteria are forming endospores and they are not inactive. Whenever these bacteria are forming endospores and they are not active, we can stain them at this stage. You can see the methods used for endospore staining. What we do is application of Malachite Green, that is the stain that we are using, and we are using heat moderate in the next step, and we are using the water, and then we are using the second stain, that is the Saffroning Stain. So there are two stains that are used. It's also a differential staining method where we are using Malachite Green and we are also using Saffroning. Malachite Green is the dye that is taken up by the endospores, and the regions which are not stained by the Malachite Green will take up the red color stain. That's why whenever we have the bacterial cultures, bacteria in a fluid or a body tissue, and if there is endospore, that endospore region will appear green in color, whereas the other region where there is no endospore will appear red in color. So that is how you can differentiate between the endospores and the non-endosporic region of bacterial cultures. The cells containing endospores appear as red colored rod-shaped structures along with an intracellular spherical or elliptical green colored structures. Endospore staining is a method of staining proposed by Dorner in 1922, which was later modified by Schaeffer and Fulton in 1933, which makes the process much easier and effective. The spores are differentially stained by using the special procedures that help the dye to penetrate more effectively in the spore wall and stains it. An aqueous solution of primary stain called Malachite Green is applied and steam to enhance the penetration of the dye to impermeable spore coat. Coming to the next method of staining called capsule staining. It is a method in which acidic and basic dyes are used to stain the background of the bacterial cells respectively so that the capsule can be visualized easily. Capsule is usually synthesized in the cytoplasm and secreted to the outside of the cell wall to surround the bacterium. It's a protective layer. Capsules are usually made up of polysaccharides, and some capsules are made up of polypeptides also. The main principle of Menneval's method of capsule staining is to stain the background using acidic solutions. For example, Congo red stain is used here, and the stain of the bacterial cell using the Menneval stain. Acid fusion being the main component of the Menneval's stain penetrates the exopolysaccharide layer with the help of phenol components. So this is the simple method of staining the capsules. What you are seeing here on the screen, the bacteria where the background is stained and you can see the capsules that are stained and the rod-shaped bacteria that are stained. The background appears white in color, which is stained. So this is called capsule staining. Here only the capsule, the outer layer of the bacterial cell wall is stained, and this is another method of staining. So that is about the different methods of staining bacteria that are normally used in a microbiology lab, and these are all the basic microbiological techniques. Thank you very much for watching this video. Please open the description box to find the details about this video. We'll meet again in the next video with some other topic. Thank you very much. Thank you for watching my videos. Please like, share and subscribe. We'll meet again in the next video with some other topic. Thank you very much.